Hello, everyone. I'm Dr. Kevin Princess, and I'm an associate professor here at Texas A M University-Kingsville. And I first want to start by thanking the conference organizers for putting together this forum where I can present my work and also accepting me to give a virtual talk here. So it's very exciting, and I'm pleased to have the opportunity to highlight some of the works that my students and I did. And the work I'll be presenting to you here in this talk here today is based on a review article that my group published towards the end of summer, this summer, beginning of fall. I think it was in August when it was published. So it was a review article based on a class of enzymes known as Flavin monooxygenases. So this a very broad class of enzymes, and all these enzymes utilize this molecule shown here as a cofactor. So this is the isoalloxazine ring of flavin. Riboflavin is derived from riboflavin, and if you know a little bit of biochemistry, these are found in FAD and FMN, which I'll be talking about here today. So before I get into that, I want to give you just kind of a background of where I'm from here in the United States. We're at Texas A M University-Kingsville. So get here. So we are not A&M College Station, the main campus of the A&M system. We're a sister school of Texas A&M at College Station. We're located in South Texas, so it's Kingsville, Texas. We're very close to the Mexican border, actually. Both Laredo and Brownsville, both of those about an hour and a half or 2 hours away. So we're very close to the Mexican border. It's a rural part of South Texas that I teach in. And we serve the chemistry department here at Tamook. We serve predominantly undergraduate students. We do have a master's program, but no PhD students. So we're not an R1 institution. And the work I'll be presenting to you here today is not, it's actually not, I'm not actually presenting any experimental work here today, even though we do some research. So being in South Texas, it's challenging in the sense that we are very kind of a poorer region of Texas. And our students are predominantly, actually, vast majority of them, 75-80% of them are first generation in college. And they're predominantly Hispanic students. So we have a lot of students that come in a little bit underprepared, but they work very, very hard. And so my goal as an educator here, which I find very, very rewarding, is my goal is to take these students that work really hard and they're really, really appreciative to be giving opportunities and to train them in basics. So I'm not presenting experimental work today, I'm presenting a review article, but I do have students that do, undergraduate students predominantly, that work in my lab, and to teach them the very basic biochemical techniques of pipetting, working in a sterile environment and all that kind of stuff. And hopefully we can train them and then spit them out and they can go on to other places. And actually the first author of the review article I'm presenting to you here today, she just got accepted to Baylor up in Houston. So she'll be going to medical school. So it's very rewarding and a very good thing to say. And I'm very proud of my students. And yeah, so the work I'm presenting to you right now is done exclusively by undergraduate students. And well, I'm very proud of them. And again, I thank the conference organizers for giving me an opportunity. So you're probably wondering what I'm actually going to be talking about because I'm going on about my students. And this isn't a recruitment talk, I realize. So just an outline of what I'll be talking about. Like I said, I'll be talking about a class of enzymes known as Flavin monooxygenases, which I'll say FMOs for short because it's easier to say FMO than Flavin monooxygenase. So I first want to introduce you to these the enzymes, where they occur in nature, and the very basic properties of them and flavin cofactors in general. And then I'll go into and talk briefly about the very broad classification. So the class of enzymes that I'll be talking about, FMOs, there's actually a lot of them, really a lot of them, about 250 or so have been characterized to date, and they could be broadly classified based, and I'll present you the very broad classification of these enzymes. And they can be grouped into three types, most broadly speaking. And this is based on the mechanism of flavin reduction. So that molecule I showed on the title slide, how it gets reduced down. So that was the oxidized form, how it gets into a reduced form of the flavin. And so they're classified based on that, and then also the subsequent oxidation of how the reduced flavin will react with molecular oxygen to end up oxidizing a substrate. So I'll present you the three very broad types that occur, and then I will also present the more specific classifications. This was done in 2014. So they are more specifically grouped into 8 groups, which there's no way I'll have time to talk about all of the 8 groups. But this classification is based on structural homology and also some of their function. And as opposed to going through all 8 of the groups, which would take quite a more than a lot of time that I have to present to you guys here today. What I want to do is just kind of highlight all the 8 different groups. I'm mainly going to be talking about the broad classification, the general mechanisms that these enzymes utilize, and then I will just highlight some of the give you some examples of biotechnical of uses of FMOs in the biotech industry. Okay, and then finally I'll end with some concluding remarks. And okay, so before I get into everything and even get into my introduction, I first want to tell you what was the genesis of this whole project in general. So it turns out about a year or 2 ago, in my group, we discovered an enzyme that we suspect to be a Flavin monooxygenase, and we've characterized it some, and we're pretty well convinced that it is an FMO. So when we were doing this, or well, when we get this, we've got a couple of publications out. I actually had last year, I submitted a grant to the National Science Foundation, hoping to get some money to fund this project. And so I submitted a grant proposal, and it came back. Unfortunately, it wasn't accepted, and I got some good comments though. And one of the biggest criticisms about actually the talk was from a reviewer. It was a legitimate comment. Was, well, the PI doesn't seem to be very familiar with the research. With the literature that's out there, because a lot of what I was proposing actually in my proposal had actually already been done. And one of the reviewers said, well, there's a whole wealth of literature on FMOs, and Dr. Francis needs to go look at that because a lot of what he's been proposing has already been done. So that naturally, after I dusted myself off because I was disappointed that my grant didn't get funded, I went in and started a search and started to take the reviewer's suggestion. And now this is a little bit of exaggeration because I didn't have a this broad of a search, but if you just type in Flavin monooxygenase into a search engine such as PubMed, you'll see a whole lot of articles will pop up. So you see a page, and I cropped this out of what I took a screenshot of typing this in, and it's page 1 of 350 or 306 different papers. So really, there's thousands and thousands and thousands of articles out there on FMOs, and they have been studied for many, many years, which I knew that, and I just didn't really go into literature that much. And the reviewer, one of the reviewers of the proposal says, the PI really needs to be more familiar with the research. So the genesis of this whole project in general was a literature search that I did, I guess, about this time last year when I got my comments back that my grant wasn't going to be funded, and getting into that and starting looking at all the different articles, it rapidly gets overwhelming. And so the purpose of I did that over the course of last winter break in the spring, and then I took on some students, and some of the students were actually, this was one of the students. Madison Guest was one of the students that was working with me in the spring and summer. So we did a literature search of this so I could prepare my talk. And but anyway, so the biggest purpose of this review article that we published, and this was in a journal article that's shown on the screen right now, was basically to go through, to tease through the wealth of like I said, there's really a whole lot of articles out there, and it rapidly becomes overwhelming if you just approach the field. And we wrote this review article essentially, if anyone, such as anyone and this is also why I'm giving the talk, if anyone in the article, anyone in the audience finds themselves in the same situation to where they suspect they might have a monooxygenase, a Flavin monooxygenase, and they need to start doing a literature search, as we put, since we were doing, me and my students, we were doing the literature search anyway, we put it together in a review article. And so this article that we published was based, it was in a biomedical journal, and we focused more on the biomedical aspects. But a lot of FMOs have have a lot of importance in biotechnology as well. And essentially, what we did is we went through all of the different classes of FMOs, and we pointed out what was the kind of the model enzyme, the best characterized enzyme. And then also in this particular article, we were pointing out the ones that have a particular biomedical importance. And we got a paper out. And like I said, this student, Madison Guest, she'll be Dr. Guest soon because she got accepted in medical school. So I'm very, very proud of her. And I just want to point out also, so if anyone's looking at this and they happen to find themselves, this is the goal of my actually talk here to you today. If you find yourself in a situation and you want this paper, you want any more information, feel free to contact me, and I can hopefully guide you into the right direction, or just help you out. I do want to point out, and this is probably more actually, this was a very good source for this paper here. This was referenced in this paper. So there's a group from the Netherlands that really the group of Van Berkel and well, some other people. They were the ones that came up with the classification of Flavin monooxygenase. They seem to be well, this group here, Frere and Van Berkel. They're really the world experts, and this is a group in the Netherlands. So this was a very good source when I wrote my paper, and also would be I think of general interest to the audience in a biotech conference, because this particular review article really talked about the difference, and it's much more longer and extensive than the ones that we published, but it talks about a lot of the biotechnological applications of FMOs. And another one here I'll just briefly mention was an Italian group. They talked about the Flavin monooxygenase as well. They also talk about oxidases, but this was another source that we pulled a lot of our information together to write our review article. Okay, so you might so I haven't really even said what the Flavin monooxygenases are yet. So what are they? Well, like I said at the very beginning of my talk, they're the F in FMO is because they utilize this cofactor shown here in yellow. This is a flavin, so this is the redox active part of flavins. It's chemically speaking, it's called an isoalloxazine ring, and it's where the redox chemistry of flavin-dependent enzymes occur. So you see in the slide here, actually Van Berkel and the group from the Netherlands, they classified these in 2014 into 8 different groups based on structural, mainly based on structural homologies and also mechanism. But like I said, when I first approached this, it was a little bit overwhelming because they have been studied for a long time. This is kind of a side note here. The very first FMO to be characterized, even though they didn't know it was an FMO at the time, was an enzyme known as lactate monooxygenase. And this was characterized way back in 1947, so almost 75 years ago. And when I started my review, like I said, in that PubMed search that I showed you, there's there's really thousands of articles out there that you can find, and there's been around 250 described. And these FMOs have not only been just annotated as a gene, looking at a gene that it might be an FMO. There's actually been 250 enzymes that have been at least partially purified and characterized to actually be a monooxygenase. And I'll tell you what a monooxygenase is in just a second. Like I said, they were characterized by that group in the Netherlands. First in 2006, and this will be the main classification I'll be talking about here today, into 3 groups, and these groups were based on how the flavin gets reduced. So what electron source comes in to reduce this molecule here, the isoalloxazine ring, to reduce it down, and then also that it subsequently so the reduced flavin will then react with molecular oxygen, and that'll activate molecular oxygen and allow the actually the organic chemistry to occur. So it was first done in 2006, and then it was updated to this the current classification of these 8 different groups, A through H. This was done in 2014, and there's a lot of very famous papers on para-hydroxybenzoate hydroxylase, which were in the late 80s, early 90s. The group from the Netherlands, they actually came through. This is the group of Vince Massey at the University of Michigan. They did all kinds of really beautiful studies on this enzyme. So this is really the very first, at least biochemically, to be studied in detail, is para-hydroxybenzoate hydroxylase. So there's a lot of studies about the flavin dynamics and all kinds of really cool studies, I guess. One thing that I found interesting in terms of when I was studying FMOs is that even after they've been studied for over 75 years, there's still, really within the past decade or even past 5 years or so, there's still papers in Science and Nature's of new chemistries actually being discovered with FMOs and flavin chemistry in general, which I think is interesting. Nature has a lot of secrets that even you could study something for 100 years and still find different things, which I found fairly interesting. Okay, so flavin monooxygenase. What is the definition of a monooxygenase? So, it has a flavin, so it does have this isoalloxazine ring, and the monooxygenase part, as the name suggests, so it, mono, 1, oxygen, 1. It takes, so what they do by definition, and I show right here just because it's probably the best characterized FMO that's exists, period. It's para-hydroxybenzoate hydroxylase. So I just showed this reaction scheme. By definition, what a flavin-dependent monooxygenase is, it's an enzyme that utilizes a flavin cofactor to oxidize, to activate molecular oxygen, shown here in red, and it takes one atom of dioxygen and it puts it into the organic substrate, as shown here, and the parahydroxybenzoate. This parahydroxybenzoate gets converted to this molecule here, but you see one atom of molecular oxygen gets incorporated into the organic substrate, and the other one gets reduced to water. So to put it a little bit more concisely, what a Flavin monooxygenase is an enzyme that catalyzes a redox reaction where it takes one atom of molecular oxygen and it puts it into an organic substrate, and it takes the other and it reduces it down to water, as opposed to an oxidase, which will reduce it down to hydrogen peroxide. Okay. So the active part of the molecule of the enzyme, is really shown here, and I showed this a couple of times already. This is the isoalloxane ring. This thing showed in red. That's where the redox chemistry happens. It's what the actually will undergo. Will get reduced. This is in the oxidized form. So this red part of the cofactor here can accept electrons to get reduced, and then it's the reduced flavin that will then go on and react with molecular oxygen to activate it, and then the molecular oxygen O2 will be active enough to where one atom can be incorporated into the substrate, the other can be reduced down to the water. So the active part is shown here in red, and I won't have time to talk about all of this, but all of this tail here is essentially, so there's different forms. There's riboflavin, which is vitamin B12, I believe, and then you have different cofactors. So there's FMN, there's FAD, and this part of the molecule is essentially there to anchor the cofactor to the protein. So these cofactors, they, in monooxygenase at least, they don't tend to be covalently attached, but they're very, very tightly associated with the proteins through mainly hydrogen bonding electrostatic interactions. And don't have any time to talk about this here today, but the isoalloxane ring, this red part of the cofactor here, has very distinct UV visible absorbance properties. And so a lot of groups such as Vince Massey, Pablo Zabardo, there's all kinds of different groups out there that can use all kinds of rapid mixing techniques to actually measure the kinetics and determine the mechanisms of these enzymes. So they're very well characterized. And through these studies, and like I said, I found it very interesting that the first FMO ever discovered was way back in 1947. So late 40s, early 50s, back I guess in World War II era. So this is the C4A addicts from the Flavin were really well characterized in the 80s and 90s. And that parahydroxybenzoate hydroxylase is one that comes up. So these are all activated. These are all addicts that form between molecular oxygen and the reduced Flavin. I mean, like I said, the C4A addicts have been known for years and years and years, but a lot of these N5 addicts and other addicts too, other addicts are still being discovered, and some have been discovered as just 4 or 5 years ago. So there's very, very rich chemistry in the isoalloxane ring of the cofactor, and that is what. So again, the overall idea here is all of these rings, all of these flavins. They started out in a reduced form, and the reduced form, this is the oxidized form, but the reduced form comes down, and the reduced form can then react with O2, molecular oxygen, and then it goes into some sort of really active. So there's an O2 here, there's an O2 an oxygen over here. So all these addicts, the oxygen gets activated, and then that can gets active enough to where the oxygen can be incorporated into the substrate and water. Okay, so like I said, in 2004, the group of Van Berkel, the group in the Netherlands, they classified these FMOs, broadly speaking, into 3 basic types. So you have single component Flavin monooxygenases, and these are enzymes that only have one subunit. So one active site, one protein molecule will carry out the entire redox reaction, the entire monooxygenase reaction, all in one subunit. And so shown here is there's a tightly bound Flavin cofactor to an enzyme active site, and that'll utilize some sort of electron source. And actually, these are 2 different groups, group A and B. They utilize a nicotinamide adenine dinucleotide reduced form as an electron source, and the NADPH or NADH, depending on the class of enzymes, will actually bind as a substrate. So there's an oxidized, a yellow enzyme that will bind the nicotinamide. The nicotinamide acts as a substrate. The electrons, the hydride, the H here, will transfer on to the flavin to take it from an oxidized state down into the reduced state, and its reduced state can then go on to react with molecular oxygen, and then that'll activate it to generate one of these addicts here. Actually, most of them do the C4 addicts here, this will activate the molecular oxygen. Molecular oxygen will then be incorporated into the organic substrate to make a product, and then the other atom will go into oxygen. And the difference between A and B is essentially the order of binding of NADPH, the binding and release of the NADPH. And I don't have time to go into details here, but this was actually a criticism that I had. That's when I first started studying on this. The reviewer said, well, Dr. Francis really needs to go and read the literature here to see what the difference are those, but that's kind of a side note here. But this is one class here, one class of enzymes. So there's single component monooxygenases. The takeaway of this is all of the redox chemistry happens in one active site. Okay, so just one component. And then a lot of other, actually the majority of flavin-dependent monooxygenases, they're actually two component systems to where there's actually two different enzymes that work together. So you'll have shown over here, you'll have a reductase component. And what that reductase component does is it'll take the flavin cofactor, either FAD or FMN. It'll take that on as a substrate. And then it'll also take some sort of electron source, and it usually is a nicotinamide adenine dinucleotide. And it'll take the electrons from the nicotinamide cofactor, and it'll reduce down the flavin. So it takes the oxidized flavin into the reduced form. And then there's all kinds of really eloquent and I think beautiful studies to where this shown in the right part of your screen right now, that would be one enzyme. That enzyme would then tightly associate with a second enzyme, and the second enzyme will be the monooxygenase component, and it'll deliver. So it starts out with an oxidized flavin, it picks up electrons to reduce it down, and then takes that reduced flavin, and then just through diffusion, there's a tight complex that will form between the two enzymes. The flavin will be transferred from one enzyme to another enzyme, and the second enzyme will be the monooxygenase component, and this component will then take the reduced flavin and then also react with molecular oxygen to catalyze the monooxygenation reaction. And there's all kinds of really eloquent studies of how the two different enzymes come together to carry out this reaction. So they work together. So you have just to recap what I said, you have a single component monooxygenase. This is where both the oxidation and the reduction occurs in all one enzyme, one protein. And then the two component of monooxygenase is two proteins working together. One of the proteins will take an oxidized flavin, reduce it down by some sort of electrons, and then take the reduced flavin and deliver it to a second protein, which will actually catalyze the oxidation reaction to convert the organic substrate into a product. Okay, and you see the more recent classification, 2014, you see actually the majority of FMOs are two-component systems like this. And then there's some other, there's another type of enzymes. These are sort of like the single component enzymes, except they don't need a nicotinamide or any sort of external electron source. These are called internal FMOs. And these internal FMOs are self-sufficient. So within the same, so the enzyme, both of these would be different enzymes. So it's still just one enzyme. It's not a two protein system that's occurring here. It'll take on an organic substrate, and the flavin will actually take some electrons from the organic substrate to reduce down the flavin, and then the same enzyme will take that reduced flavin and react with a product. So it doesn't need an extra. Well, the reduced flavin will react with oxygen and convert it into the reduced flavin will react with molecular oxygen, transfer one of those oxygen atoms into the substrate and the other into water. And these, so the only difference between the internal monooxygenases and the single component monooxygenases is that the single component monooxygenases actually have an external electron source, usually a nicotinamide, whereas these internal monooxygenases utilize the organic substrate. Well, it takes electrons from the organic substrate and essentially puts it to another part of that substrate to convert it into product, and it's all done by itself. It's self-sufficient. So I'm running out of time here, and I just want to, like I said, I'm not going to go through the initial classification of FMOs in 2004 was just based on those three type of mechanisms. And then as these FMOs have been explored, especially now that the X-ray crystal structures are much more prevalent than they were even in 2004. The same group went and classified a little bit more A through H. And I don't have time to go through all of the different groups, but if you're interested, you can go to the paper, my paper, or another group's paper, and look at all these all the different types of enzyme groups A through H. What I did here is I just want to highlight some of the biotechnology. Well, just tell you there's a lot of applications of FMOs in the biotech industry. And so I just want to highlight a few of them. So group A, like I said, this is the best characterized group, or the first group to be extensively characterized with para-hydroxybenzoate hydroxylase. There's a lot of them, including this one. This is rifamycin monooxygenase that is involved in the production of antibiotics, the production and degradation of antibiotics. There's all kinds of different applications. So you can use these FMOs to generate antibiotics. They're also widely used in biocatalysis. The group B enzymes, I just showed one example here, which I believe is SID-A. These can be used in the biocatalysis, different biocatalysis reaction to generate all kinds of different useful products of antibiotics and other things. Well, I do want to say this. This is probably the most famous FMO known, which is luciferase. Luciferase is widely used in molecular biology techniques because it actually fluoresces, and it's a very useful and convenient enzyme to use to report gene expression. So a lot of times when genes get turned on, luciferase will actually glow, well, not glow, but it'll fluoresce, and you can see when genes are turned on and turned off, and on some sort of application and molecular biology or something like that to tease out diseases and disease processes. So that pretty much ends my talk here. So again, I want to thank the conference organizers for giving me an opportunity to present my work. And I hope I showed that after 75 years of studying this class of enzymes, things are still being discovered day to day. So it's worth studying, and it can be rapidly overwhelming to go through all the literature. But hopefully, if you go to some of these reviews here, you can tease through and see. Well, hopefully my talk illustrated that the very broad type of mechanisms that exist, and that's pretty much all I have. So I do want to acknowledge our funding sources. So we are funded by the Welch, our department's funded by the Welch Foundation. My college has also been generous in providing some research support, and we got some support from the McNair scholarships and some other funds. And I thank everybody and the conference organizers once again for giving me an opportunity. And thank you very much.